So let's make a start on our first Unit 3-4 topic. The very first thing we're going to be covering then is kind of an introduction to how cells and how organisms work. And for that, we need to look at the topic of biomacromolecules. Now, straight away, one thing I'm going to tell you for Unit 3-4 is you're going to get quite a few big words like this throughout the year, quite a few technical words. Maybe you've seen the subwords before, but it's highly unlikely you've seen them combined in this way. But whenever you see a word like this, right, there's a process you want to follow to understand what's actually happening. Right, you could just memorize the definition, but if you understand where this word is coming from, it will make a lot more sense and help you to remember. So the first thing we want to do is always break it down into the subwords or root words that you actually recognize. So for biomacromolecules, I'm going to split that down into biomacromolecules. Now I want to go into Latin and then derive what is this actually saying. So all these words usually in biology or any scientific terminology, they're always derived from Latin or they have a similar word in Latin. This example is probably a bit more straightforward for English, right? Molecules, I hope we all know what molecules are. Macro is just talking about on the big stage. And then biology, or bio for short, is going to be talking about life. So what this is saying, molecules, or if you can think of like atoms or the substance, what are the big atoms? What are the big substance that is responsible for making up life? So this is kind of the process we will always follow for breaking down and understanding what these words mean. So on a similar way, can you guys try? I'll give you 1 minute. We have two words here, genome and proteome. Can we try and first do the first step, break it down into what words can you recognize? If I give you a hint, home is a word in Latin that means set. So what else is left?
Gene.
Good. So we have got gene in the first one, and hence what's going to be the second one?
Protein.
Protein. So genome is just the complete set of all genes. Usually it's going to be in an organism. So you might be talking about it in the context of a human, or you might be also talking about it in the context of plants later on. And hence proteome, by definition, is the complete set of all proteins. So the first topic we'll be exploring though is biomacromolecules, and we'll come to genome and proteome later in this booklet. For biomacromolecules, there's 4 key ones, and each has some important properties and kind of roles that we need to be aware of. So first biomacromolecule, right, protein, which we just started with. We also have what makes up a gene, which is going to be nucleic acids. So these two are our most common biomacromolecules. Protein is the most significant one. We'll be doing a lot about it. But nucleic acids is also up there in terms of bio 3-4. The other two, we have carbohydrates and we have lipids. Now, if you've done Unit 1, 2, you might remember covering this in a little bit more depth, but we don't really do it in Unit 3, 4. There's only a few things you need to know about these. The firstly is that carbohydrates is going to be a source of energy. So in the context of humans, we're usually going to be talking about glucose for this. And for lipids, right, this is, think of like fat or fatty acids, or if you know glycerol, if you've done a little bit of chemistry, right? But it's the substance that made up our phospholipid bilayer that we covered in our Unit 1, 2 revision. So phospholipid, again, if you break that down, phospholipid bilayer. Phospho just means phosphate group, right? Again, phosphate group is a chemistry term. I don't expect you to know that, but you can just understand that it's a lipid with a phosphate attached. So if we know it's going to be part of our phospholipid bilayer, one of its central roles then is controlling the semi-permeability of cells. Now, for both of carbohydrates and lipids, we should also know where they come from. So carbohydrates, they are usually going to come from the diet, right? So you need to consume them. Whereas lipids, again, back to our Unit 1, 2 revision, is going to be made in the smooth endoplasmic reticulum. Endoplasmic. So in terms of carbohydrates and lipids, that's pretty much all you need to know. There's not a lot there. Proteins and nucleic acids, I will leave for later in the booklet. So then I want to cover the aspect of what is polymer and what is a monomer. So again, if we apply what we just discussed at the very start, we're going to break down the words into the actual meanings. So polymer can be split into poly and mer. Monomer can be split into mono and mer. Now, if you derive what they mean from Latin, poly just means multiple. Well, so I have multiple. Mono just means one, and mer is equivalent of unit. Right, so a monomer is one unit. A polymer is multiple of those units. So you can imagine that if we're talking about biomacromolecules, right, so really, really large molecules, those are examples of polymers because they consist of lots of units put together to form kind of this really long chain of repeating subunits. Now, what are those subunits? Those subunits are the monomers. So if you imagine we're building a protein like this, right? This protein is a polymer. So a polymer, what was it? Repeating subunits. Of a monomer. So then if you take one individual block, this is what we are calling a monomer. But a good analogy to remember, right, you can think of monomer as a single train carriage. You can think of a polymer as the entire train. So when you join together, trains aren't too long, but if you imagine you have to join together 100 or 200 of those monomers together or individual train carriages. That's how you can form the complete train overall. Now, for each of these biomacromolecules then, right, they're going to be all of these, are polymers, which means they're all going to have the monomer subunits. So for protein, right, if protein is the polymer, its subunit or its monomer is going to be an amino acid. If we're talking about nucleic acids, its subunit is going to be a nucleotide. Now, lipids, these are kind of an exception to the rule. They are technically like very similar to a polymer, but they have two monomers, not just one monomer. So they're actually made up of two like repeating subunits, one after the others. So it might look like this, this, and then it goes back. If you imagine red and green are representing different molecules. So for this reason, we don't call a lipid a true a true polymer because it's made up of those alternating monomers. Hence, the very last one then is carbohydrate. And its monomer is a monosaccharide. Which is a very fancy term for the simple sugars. So again, think glucose. Now, it's very good or it's very important for you to know at least these three. If we want to ignore lipids, that's fine. But we need to know what is the monomer and what does it make up for the biomacromolecules. Okay. So that's a very quick intro, I guess, as background. For each topic, there will kind of be like a summary page here, which is really good in terms of revision. So when I talked about when you should go back and revise the booklets, what should you actually specifically be doing? On like the first revision, it might be a good idea to watch the recording. But if you're coming back for the second or third time, I don't think it's going to be worth it anymore. Then these summary pages that come in are the key areas you really need to be going to first for content. They're really good because they break it down into the key characteristics. They've got the keywords, right? So if you're writing an answer to any sort of response, the more of these keywords that you are using, right, the higher the likelihood is that you're actually answering the question correctly. If you've written an entire response and you've used 0 of these keywords, the chances are you've probably gone a little bit off topic, off track, and even if you actually have hit the right idea, the examiners really are looking out for these keywords to give marks. So you might still be missing marks without using keywords. Because I'll talk you to like a VCE examiner. They have around 5 minutes to mark each paper. So you're dividing 120 marks by 5 minutes. Calculate how many seconds you can get per mark. It's not too many seconds. So they don't really have the time, if you've written a whole paragraph response, to read through that. What they're going to be scanning for are these keywords. So that's another reason as well to include the keywords. Not only does it show you on track, but the examiners will be scanning for them to award you marks. For SACs, it will work a little bit differently because your teachers are more likely to read the whole thing, given they have more time and less students. The examiners are very, very quick at marking exams And so they really rely on keywords to help them market. Another good thing it also has is a reference to a past question and what students have done wrong or what was tricky about that question. So it's kind of like an extension, particularly if you're aiming for 45 plus or even 40 plus. It's really, really important. You read this, it'll be given for every subtopic, and it's like the most common way that they like to trick you in that subtopic. So again, I think we talked about this just here, but it's repeated. Lipids are often discussed alongside these, but they are not true monomers because they don't have the same repeating single subunit monomer, right? They're made up of two different things. Okay. One more thing to do in the review as well, sorry, is to go through the questions. Right? Particularly, maybe in the first 3 times or so you're coming back to review, you should write out your answer and then mark it. Every single question has a marking scheme attached to it, as well as a model answer, as well as common mistakes to avoid. Right? So it is very comprehensive. Obviously, we'll go through it in class once, so you can watch the recording. But even if you don't want to go through it, right, all the information you need will be given for every single question in the booklets. So this is one thing that should definitely, or like the most important thing to do in the review. If you're coming back to the revision for like the 4th or 5th time, and you know you can constantly get 3 out of 3 or 4 out of 4 marks, right, that is the point where maybe you don't need to write out your answers. You can just think about it or think about the marking scheme. But in your first couple of times, right, it's a very common, I'm not really sure it's like any arrogance or even something that students think they can just do the question in their head. Yes, you can do that very later on when you like are super confident with the content, but please don't do it at the start because that makes it very difficult for you to assess yourself. And then if you want to send the question to me or Neil, if it's done in your head, obviously we can't grade that either. Okay. Sorry. Any questions so far? No. Okay. So let's move on to the content. The first thing then, we'll actually dive into nucleic acids. So remember, this is one of the biomacromolecules, and its monomer was a nucleotide. Now, we have two types of nucleic acids, DNA and RNA. They have 5 important differences between them, and this is a whole year thing, right? So every topic, like DNA versus RNA, comes back up in evolution. It comes again in immunology. It comes again when you do cellular respiration. And obviously, it's here in the first topic. So it's a very, very fundamental concept that comes every single area of study. So what actually are those 5 differences? Firstly, DNA, it is always double-stranded. RNA is single-stranded. Now, I'm going to put an apostrophe on single-stranded for RNA, though, because this is usually, right? So there are a few exceptions for RNA where it can be double-stranded. So what that means is if you're not told, right, let's say you have a diagram and you're not told it's DNA or RNA, you've just got 2 strands. You don't want to use the fact that it's double-stranded to assume that it's DNA straight away. You want to go to one of these other differences. So, for example, the second difference, the sugar, So we will look at the structure of DNA and RNA in the next page, but sugar is one of the components that makes up DNA and RNA, and they have different types of sugars. So DNA has deoxyribose sugar, whereas RNA has ribose sugar. So that is an example of a concrete difference. There is no way or no scenario where the sugars can be flipped around. So if you see this strand has deoxyribose sugar, you know for certainty it has to be DNA. Uh, One thing about its full form as well, given that we covered that sugar is deoxyribose, what DNA actually stands for is just deoxyribonucleic acid. Right, so nucleic acid we discussed. DNA is a type of nucleic acid. What type? It's a deoxyribo type. Then that's based off the sugar. So that means you can probably guess what RNA is. Sugar plus the biomacromolecule, hence ribonucleic acid. Okay. Next, we have the nitrogenous bases. You can see that's the next topic. So the bases we have for DNA is going to be, oops, A, T, C, G, and for RNA, we have U, sorry, U, A, C, D. Now, in terms of the location or where is it found, right, DNA is always going to be found in the nucleus. RNA is going to be found in the nucleus plus the cytoplasm.
I have a question.
Yeah,
Uh, didn't you say like that the mitochondria and the chloroplast had like circular DNA or something in them? Remember, like I think it was like a few lessons ago because I watched the
yes, that's
report.
right. So you will find DNA in the mitochondria and chloroplast. And it's very good that you picked up on that. It's often going to be tested in MCQs. Uh, However, for area of study one particularly, we are focusing on what we call the protein secretory pathway, or how do cells export proteins to the rest of the body. For this particular pathway, the DNA has to come from the nucleus. Later on in the year, particularly when we get to cellular respiration and immunology, that is when we'll be focusing on DNA in the mitochondria and in the chloroplast. And why does this cause other differences, or how is it different to the DNA in the nucleus? So you're absolutely correct that DNA is found there. But for this particular topic, our emphasis is DNA in the nucleus. Chloroplast. And lastly, we have the lifespan. DNA basically doesn't degrade, right? The DNA you have is from birth, and you will have it all the way, even like 1,000 years after you die. So it's a very, very long-term molecule. In comparison, RNA is a very short-term molecule. Usually it will be produced and then destroyed within around a few days. So these four here are all concrete differences, right? They will always be there. So these are the ones you really want to use as proof to identify if something is DNA or RNA. In terms of the strands, right, this is just more usual. So usually DNA is double-stranded, usually RNA is single-stranded. but there are a few exceptions, and so that's why we don't like to use it as a gold standard proof. Okay. Then if we have a look at the nitrogenous bases, so what actually is ATCG that I'm talking about? ATCG is standing for these molecules we have given here. So A represents adenine, G is guanine, C is cytosine, T is thymine, and U is uracil. You see you have the chemical formulas above. You do not need to know these chemical formulas. Way too much chemistry, we're doing biology. What you do need to look at in the difference, though, is the rings. So you can see cytosine, thymine, and uracil, these are all single rings. Whereas adenine and guanine, you can see they are double rings. 2 rings versus 1 ring. Now, if you have a 1 ring nitrogenous base, that is given the name pyrimidine. If you have a 2 ring nitrogenous base, that is given the name purine. So, every nucleic acid molecule is going to have a nitrogenous base. Now, both DNA and RNA have 4 options. A, C, G are common, but for DNA, we have thymine. So DNA is thymine, and RNA, we have uracil. So uracil will replace thymine for RNA compared to DNA. This terminology, again, is something they like to test in MCQs, but it doesn't hold much further reference. So it's just a kind of definition to know for an MCQ. In terms of the 6 plus 5, again, you don't need to know how big the ring is. Just memorize 1 ring versus 2 rings. That's all that's important. Okay. So now that we've covered nitrogenous bases and what they mean, we can look at how do they go together. So what we have is this idea of complementary base pairing. What that means is the topic we just called purine and pyrimidine, they go together. So a purine will bond with a pyrimidine. Now, there's a little bit of chemistry here, so I'm not going to go into in too much depth. But if you have a look at the differences in structure for guanine here and adenine here, and you compare it to cytosine here and thymine here, but guanine is only able to bind to cytosine because of those chemical bonds. And adenine is only able to bind to thymine or to uracil. So what our bonds are is that adenine pairs with thymine for DNA or uracil for RNA, whereas cytosine always pairs with guanine. So this idea that one specific purine will always bind with one specific pyrimidine is what we call complementary base pairing. Now, this idea is very, very important because once we look at double-stranded DNA, right? This is strand 1, strand 2. How do you connect 2 strands of DNA together? You connect them via complementary base pairing. So if this strand of DNA has adenine here, the other strand of DNA must have thymine here, and then they can bond together. Does that make sense? Any questions so far? Okay. So let's actually draw the structure of nucleotides, right? So we've talked about some of the subcomponents. I've talked about sugar. I've talked about the nitrogenous base. How does this all actually fit together? This is a really good point to draw the diagram with me as well. That will help to reinforce your learning. So a nucleotide has three major components. But one, two, three. Now, the nitrogenous space we are talking about is this rectangle here. The pentagon we are talking about represents the sugar. And finally, then, the circle at the very top represents the phosphate group. So that's the same phosphate group we were talking about for phospholipids. You do need to follow these same shapes, right? So it's universally accepted that circle means phosphate group, rectangle means nitrogenous base. You can't switch the shapes around or try to use different shapes. You have to use these three same ones. It's also important in your diagram that these lines we're drawing off to connect the nucleotide together or the subsets together, it needs to come from this corner of the pentagon and this corner of the pentagon. Right, so you cannot draw the line from here. That is incorrect. It must come from this particular corner, right? Not from the one above or from the one below. It has to be that specific point. The reason behind that is we assign the carbon number to all the carbons. So this is what we call carbon number 1. This corner here is carbon number 2. This one is carbon number 3, carbon number 4, and carbon number 5. So the nitrogenous base is always connected to carbon number 1. And the phosphate group in the nucleotide is going to be coming off carbon number 4. So they need to be like well aligned. It's a good idea to even bring a ruler into your sax if you know you're going to be drawing something like this. Now, in terms of the bonds themselves, this bond here that's connecting the phosphate group to the sugar, this is termed a phosphodiester bond. Phosphodiester. The bond that is connecting the nitrogenous base with the sugar is termed a glycosidic bond. So what we've drawn now is a basic nucleotide. However, if you remember, I said for nucleic acids, it's the polymer, nucleotide is the monomer, but you have two types of nucleic acids. You've got DNA and you've got RNA. And therefore, you also have two types of nucleotides, one for each monomer. So that's going to be called the DNA nucleotide and the RNA nucleotide. If we are talking about the DNA nucleotide, there's only one thing that you need to change. That is the sugar, right? So we can't just call it a sugar. We need to specify that it's a deoxyribose sugar. If you are dealing with RNA, what is it? It's going to be a ribose sugar. If it is unspecified, you will call it a pentose sugar. So three terminologies, depending on if you're talking about DNA, RNA, or general. Now, pentose, right? Think about the fact that it has 5 carbons, right? So penta means five. Ose is, you can think of it as referring to carbon. That's not quite the translation, but it's basically saying it's a sugar with 5 rings of 5 carbons. Now, if you look at the difference between DNA and RNA, ribose sugar versus deoxyribose sugar, can anyone tell me what is this prefix deoxy? What does that mean?
It means like a lack of oxygen, right?
Yes. So D means lack of, oxy is standing for oxygen. So DNA or deoxyribose is just RNA with one less oxygen. So at the bottom here, you always have, let's say this is your carbon group that we've been numbering. You usually have H, or your other option is to have OH. So RNA will have OH, and deoxyribose is going to be lacking one oxygen. And so you call it deoxyribose. So this would be DNA, this would be RNA. It's only for one carbon, not all 5 of them. Now, one thing you need to be very careful of when you've been asked to draw a nucleotide is to make sure that you fully understand which nucleotide you are drawing. They're not always going to tell you that I want you to draw a DNA nucleotide. I want you to draw an RNA nucleotide. They can be quite tricky here. So as an example, from the Haileybury 2022 SAC, right? So a real SAC, what they said is that you want to draw a nucleotide that is in the cytoplasm. Now, cytoplasm doesn't immediately tell you anything with DNA or RNA, and a lot of students just drew the general form of a nucleotide. But if you remember, when we covered the differences for DNA and RNA, right, DNA is not going to be in the cytoplasm by itself. RNA is what's in the cytoplasm. And so actually, they wanted you to draw an RNA nucleotide, which meant you had to label the sugar as a ribose sugar, not a pentose sugar. Does that make sense to everyone? Yeah, good. So some of the other ways then that they have also asked you before in SACs or in exams, I've listed at the bottom. Double-stranded versus single-stranded. You usually will go with the default, aka double-stranded DNA, RNA single-stranded. But this one is a bit iffy because, as I said, there are exceptions. So this one was tested in a SAC. I don't fully agree with testing it like this, but if it does come up, just go with your default options. Double helix, right, is referencing the structure of DNA. Right, RNA might be single-stranded like this, but if it's a double helix shape, so helical means like this sort of spiral, that has to be DNA. So this is definitely telling you DNA. This is definitely telling you RNA. They could also tell you the sugar as well. Right, so we've got a nucleotide with a pentose sugar or with a deoxyribose sugar. that example is usually a little bit easier. So that is our monomer for nucleic acids. Now, if we join it together to get our polymer, so let's draw one. Okay, so now I've drawn one strand of my DNA molecule. As we remember from earlier, this is a phosphodiester bond. Because it is joining the phosphate group to the sugar. But anything that joins phosphate group to sugar is called phosphodiester. So if you look at the bond here, right, although it's two different nucleotides, right, in the polymer, it is connecting one phosphate group to one sugar. And therefore, it is also a phosphodiester bond. So both of these are phosphodiester bonds, even though one is within a nucleotide and the other is connecting two different nucleotides together. Now, again, though, this particular way that I've drawn it, connected to this corner, is very, very important. You must connect it to that exact corner, because when we label our carbons out again, 1, 2, 3, 4, 5, and I'll label the one underneath, 1, 2, 3, 4, 5. What you can see is that the fifth carbon of one nucleotide is always going to connect to the third carbon of the other nucleotide. So if we draw it in descending order, right, what's happening is that we have a 5' here. Prime just means carbon. And you have a 3' here, because I'll just label this 1, 2, 3, 4, 5. 3 is where the next one would attach. And 5 is where it starts. And so we call this order here 5' to 3'. And that is how you always read DNA. You always read it from 5' to 3'. So this is just one strand though. On the other side, we'll have the other strand. So again, if we go to work labeling the carbons, this is going to be carbon number 1, 2, 3, 4, and 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Now, if I draw the arrows here, this is 3, this is 5. So we've got 3' on the top, and you've got 5' on the bottom. However, you cannot draw the arrow like this. This is wrong. Because we always read DNA from 5' to 3'. So you must draw the arrow going like this. 5' to 3'. That means the arrows are going in different directions. Now, if they are going in the same direction, right, this is a mathematical term, but you can call those lines parallel. Now, for biology, right, they're going in different directions. So rather than saying they're parallel, we say the DNA strands are anti-parallel. So parallel because they're still aligned, but anti because they're going in different directions. So now I've finally got two strands of DNA coming together. However, we haven't connected them together yet. So what we need to draw is our last type of bonds. Now, let's say we've got A here, we've got T here. We have C here, we have G here, and we've got G here, we've got C here. So you can write C here or on the other side, that doesn't make a difference. Now we can do complementary base pairing. And connect these two together. The bonds that connect the nitrogenous groups together are called hydrogen bonds. Hydrogen bonds are relatively weak, whereas phosphodiester bonds are strong. So that means when you try to break apart DNA, the first bond that will break is the hydrogen bond. Now, the individual nucleotides, therefore, do not break apart. All that you'll be doing is separating the two strands of DNA into individual single strands. When these hydrogen bonds are connected, though, you get the very typical DNA structure. Remember what we called that structure? Double helix. So double helix and anti-parallel are very important ways to describe the structure of DNA. And when you're talking about direction, 5' to 3' is also very, very important. So 5', remember, is always the start, and 3' is as you're traveling down. Now, what this results in is that if you want to add a new nucleotide to the group, right, so I want to draw another nucleotide, I cannot draw a nucleotide here. That is wrong. I have to draw a nucleotide at the bottom here, because the new nucleotides will always be added on the 3' end. Right, this is where you're reading downwards. This is where you can extend the downward line. So you read it from 5' to 3', and if you're going to extend it, you extend it on the 3' side. So overall, that forms your structure for DNA. Right, again, you've got the key takeaway notes. Two very, very important words to describe the structure: anti-parallel and double helix. And very important to note direction as well, 5' to 3'.
Excuse me.
Yep.
I have a question. So since you said that you can only add down on the left strand, would you go up on the right strand because it goes from 5 prime to 3 prime? Like when you're adding nucleotides on the right strand, the one that's going that's the one that's one that is anti-parallel, it's going from 5 prime to 3 prime, would you add up?
Yeah, so
Because you would
it's a very good question. One thing to remember is that your DNA is fixed from birth, right? So let's say you're born with this length of DNA. You're never going to add new nucleotides to this DNA, right? All the genes, all your genetics are done from birth. So when I'm talking about adding new nucleotides, what we're doing is forming more DNA, aka think about mitosis, meiosis. We're creating more DNA for new cells, not extending the original DNA. So what would happen for replication to occur is that you take one strand of DNA that is already in full length, and then you'll be building another strand on top of that. So the strand you are building must go from 5 prime to 3 prime. But if this represents a full length of DNA, right, you can't extend 3 prime here and 3 prime here because they wouldn't line up. So once DNA is fully formed, you can't add more nucleotides. It's in this growing phase that we're talking about, where you're adding nucleotides on the 3 prime or this side. Does that answer your question?
yeah, I got you.
Yeah, good. Okay. So again, quick summary. What did we cover for DNA? Right. Again, full name, deoxy, lack of oxygen, ribo, you can ignore that. It's not worth really. And the nucleic acid is obviously the biomacromolecule. It's going to carry genetic instructions. I should probably add from the nucleus. Two most important words that you should learn today is antiparallel and double helix. One terminology to be aware of, we will actually cover this in immunology later in the year, but I'll just give you the word now. Is a nucleoside in comparison to a nucleotide, right? We've only studied nucleotide here, right? So that's all I want you to know or be very confident in. But you need to get the spelling correct. Usually biology is not too picky about spellings, but in specific cases like this, where you've got two words that are very, very like closely related or just one letter difference, VCAR won't accept any deviation in spelling. So nucleotide is one word that you must spell correctly. Another word also from today that you must spell correctly, if we go to our basis, is adenine. Spell correctly. The other ones, right, if you miss a letter for guanine or cytosine, Vika will still give you the mark, but adenine must be spelled correctly. And same for thymine. And that's again because there's molecules that are in the body that are very, very close to it. If you are interested in what a nucleoside means, it's just the sugar and the base. So it's without the phosphate group. That's usually because you detach the phosphate group or you break the phosphate group to generate energy. You don't need to worry about that too much. We'll cover it when we get to immunology. And same thing, right? Students, common mistake. Please specify what is the sugar Is it deoxyribose? Is it ribose? Is it pentose? Sugar alone is never going to get you the mark. I think, yeah, the rest you can like to read if you want to come back to it. This tip here, again, we did cover it. Just make sure you can't make the assumption that RNA is single-stranded. It is mostly single-stranded, but not always single-stranded. Okay. So now that we've covered DNA, let's move on to proteins. Now, when you think of any function in the body, most likely it's going to be powered by proteins. So a lot of the things that are done in your body is done by proteins. So 7 particular functions. Right, first one, enzymes. What are enzymes? They are organic catalysts. This is actually a full area of study. So we will look at them in a little bit more depth, but hopefully you should know that catalysts mean they will speed up the rate of reaction. So organic catalysts mean they will speed up the rate of reaction in the body. Right, and they're naturally produced by yourself. Next function, transport. We did look at examples in early unit 1, 2 of panel proteins, carrier protein, right? So think back to those proteins. What were they doing? But they are allowing something to cross the semi-permeable membrane. Structural is self-explanatory, right? Supporting your cell structure, supporting your cell shape. Not too concerned about that one. If you have done unit 1, 2, you will hopefully know about hormones, and you should have done a lot about the whole pathway, right? How is it secreted? What are its effects? It's not very relevant to 3, 4, but linking it back to what you have already done, they are a very good example of a protein, though. And they are secreted en masse. So we will have a look in immunology at some cells that secrete proteins en masse as well. Immunology, I've been talking about that a lot, and here we go. Right, one function, again, done by proteins. Right, so there is an adaptive pathway to fight infections, and that is basically all proteins. Again, don't worry about that too much now. That's like a term 3 content. Motor contractile, right? Probably the most intuitive one. Think of any movement involving your muscles. How are your muscles contracting, or how are they extending? Again, this is all the work of proteins. And lastly, storage. Again, pretty self-explanatory, right? This just stays in one spot in the body and stores something. A really good example of this is iron. Oops. Probably one of the more common molecules that would be stored. The name of the protein that stores it is ferritin. And I'm not sure if you've read any news or done any research on health, but a lack of ferritin or iron deficiency is a very common problem in a lot of people, particularly females. Now, these are 7 functions given here. You don't need to memorize every single function. Usually, they will test around 3 functions. So you should be able to recall at least 3 functions off the top of your head. In terms of specific examples of a protein that comes from that function, you should know one example of an enzyme. Now, If you have done Unit 1, 2, you should have done the enzyme amylase. Otherwise, don't worry, in Units 3, 4, we will do a lot of examples of enzymes throughout the entire course. One that we will cover next lesson, if you want to write it down, is going to be RNA polymerase. Very quick tip, anything that ends in ase means enzyme. So you should know one example of an enzyme, and then you should know one example of a protein that is not an enzyme. Right? It can be from any of the 6 different functions. I personally went with ferritin, but you can read through the examples. There's a lot of easy ones. I think structural is one of the easy ones as well. Collagen, elastin, keratin. Like hopefully you've heard of those before. They are commonly used. Hormones is also a good one. Insulin was covered in Unit 1, 2, but you've definitely heard of that before. Adrenaline as well is an example, right? What stimulates the fight or flight response. The others, I think, like in function of motor, like protein or transport, become a little bit more difficult to remember. If you have any others that you can think of that are not examples, also feel free to message me and I can confirm if they work or not. But yes, overall, this is like a list of the 7 functions that proteins do. And so you can see they're very, very important to the entire human body. So then if we move down to have a look at the structure of a protein, can anyone tell me from earlier in the lesson, protein is the monomer, sorry, protein is the polymer. What is its monomer?
Amino acids.
Amino acids, good. So protein is going to be the polymer. Amino acid is its monomer. So we'll start with drawing the structure of a single amino acid. Compared to just the 3 parts for a nucleotide, amino acid has 5 subsets. Firstly, we've got our central carbon, but that's just the carbon at the very center. Now, to the right of the central carbon, we're going to get our carboxyl group. All right, these all in different colors. Carboxyl group, central carbon. To the left of the central carbon, we have our amine group. Amine group is, again, something you need to spell correctly because in chemistry there is also an amide group, but it's a very similar spelling that for amine needs to be spelled correctly in bio. To the bottom of the carbon, we have our hydrogen. You can call it hydrogen atom, hydrogen group, or just hydrogen. Doesn't really matter too much. And finally, to the top, we have our variable R group. You might also hear the name variable Z group. Again, synonym, same name, use whichever one you prefer. So that's our overall structure for an amino acid, 5 groups. You do need to draw it exactly like this, right? So you can't switch the amine and carboxyl groups, right? Carboxyl is always on the right, amine is always on the left. There's no exceptions to that. In terms of the variable R group though, right? Think about how the nitrogenous bases could switch around. That's the same thing for the amino acid. the variable R groups change from one amino acid to a different amino acid. In total, you have 20 different amino acids. So that means, right, 20 different variable R groups. Now, hopefully you know a few descriptive words from Unit 1, 2, and we did cover some of them in the revision, but words are like polar or non-polar, charged, right, quite literally, does it have an electrical charge or is it electrically neutral? Uncharged, hydrophobic, hydrophilic, right, so hydrophobic, hydro is water, phobic is hate, so water hating, or hydrophilic, philic means love or water loving, right, so two opposite sides. All amino acids have different properties on these three groups, and that depends purely on their variable R group. But even the size depends on variable R groups. So some can be very, very small, and some can be like close to double or triple the size of other amino acids if they have a really, really large variable R group. Do I have more space? Yes. Now, some like aspects or characteristics we should know about those amino acids. Firstly, right, as I mentioned, 20 different types split into 9 and 11, 9 essential amino acids. Now, essential, like in everyday vocabulary, sounds like you need it to survive, and you then might think non-essential means you don't need it to survive. That's not quite how we're using it in bio. For biology, 9 essential means it's not produced by the body, and non-essential means it is produced by the body. So when we say essential, we're saying it is essential to get from the diet or from your consumption, right? And therefore must be consumed. Now, that means there is 1 and 2 dot points. So mostly in biology, when I ask you to define a word or to get a definition, it's a 1 mark. However, when you are defining essential amino acids, it's a 2 mark question because you need to raise both of these points. Firstly, that it means it is not produced by the body, and therefore you must get it via your diet or you have to consume it to acquire that amino acid. Whereas non-essential is just produced by the body, so you will get it automatically. It's not necessary to get from the diet. You might get more from the diet, but your body can supply it. Okay. Any questions so far? No. Great. So if you scroll down to the next page, and I do want you to have these 2 pages up on your screen as well. I have a list of all of the 20 different amino acids. Don't panic straight away. You do not need to memorize the chemical structures of any of them, right? I'm just showing you here so you can see what is the different variable R groups I've been talking about. So if we break it down, right, here is our central carbon. Here is our hydrogen. Here is our carboxyl group. Here is our amine group. Same structure, right? Carboxyl to the right, amine to the left. Hence, anything left at the very top is always representing our variable R group. And you can see that if we compare the first two, alanine and arginine, that these two have very different variable R groups. Therefore, they will have very different properties. As an example, there's one, or alanine is very small. It is non-polar. It is non-charged. Arginine, right, is comparatively a lot larger. So it's probably not going to pass through the plasma membrane, being so large. It also has nitrogen, which makes it a polar molecule. And remember, lipids are non-polar. Non-polar will repel polar, and therefore arginine will definitely not pass through the plasma membrane. So just because of that difference in the variable R group, they result in very different properties. Now you can have a look through the different variable R groups as well. You can see aspartic acid here has two carboxyl groups, so it's got a repeat. This one is obviously the more important one. I don't think they have been labeled if they've been charged or not. That's okay. Now, one task I want you to do, can you go through these 2 pages and tell me which amino acids have sulfur in them?
There's something called cysteine, right?
Yeah, that's one of them. So sulfur is represented by the S, by the way, for its chemistry.
Methionine.
Yeah.
Not sure how to say it.
Methionine, that's okay. For any amino acid, by the way, for biology, you are allowed to abbreviate it to the symbol given here. So you can just write MET instead of having to write all of methionine out. Now, these two are the only ones with sulfur inside of them. However, if you look at the sulfur for methionine, right, it is surrounded by carbon and hydrogen groups. Whereas the sulfur for cysteine is at the very end of the molecule. So the sulfur for methionine is not available for bonding, right? If you imagine this is sulfur here, it is already crowded by carbons and hydrogens next to it. So it cannot form another bond. However, for cysteine, the sulfur here, it's only partially surrounded because it's at the end of the molecule. And so it can form new bonds. Why this is important is because we have a type of bond called disulfide bridges. Now you can see sulfide is in the name of this bridge. So to construct this bridge or disulfide bridge, you need to have the sulfur molecule. Now, in the MHS SAC, right, is a very brutal question but they gave a chain of amino acids, and they asked, is it possible to make a disulfide bridge? That chain of amino acids did not have cysteine in it. Now, out of 20 different amino acids, cysteine is the only amino acid that has a free sulfur group, or a sulfur group that can bond. And so if you want to form a disulfide bridge, you must have cysteine, or the amino acid cysteine, in your group, because otherwise you don't have sulfur to form a disulfide bridge. Now, one more amino acid trait, good one to know, is glycine. Glycine. The reason I like to would like you to remember glycine is because it's the simplest amino acid, right? So its variable R group is just a H. There's nothing more complex than that. So if they ask you to draw and label a specific amino acid, right, you can just go to glycine as your specific one. They will never ask you to draw and label more than one specific amino acid. So just remembering glycine is enough to fulfill that question type. Yeah. So that is our monomer for amino acid. Any questions on that so far? No. Great. Okay. Then if we go to look at the polymer, how do we connect the amino acids together to actually get a protein? So what we're doing now, remember, is taking one monomer, another monomer, and connecting it together. And that's how polymers are formed. You got to join multiple monomers together. So if we draw out one amino acid, I draw a second amino acid. So we'll get two distinct amino acids. Now, the hydroxyl, so the OH on the carboxyl group from this amino acid, and single hydrogen from the amine group on this amino acid are going to combine together. So if they combine together, what is that? Two hydrogens, oxygen, H2O, or that's water. So we secrete H2O. Now, the bond then, carbon and nitrogen, right, they will bond together. So if I draw how that looks like, carbon, carbon, oops, sorry, nitrogen, right, hydrogen goes to the top. Oh, sorry, I drew one less oxygen here. Did I draw it correctly? Yeah, I drew it correctly there. Oxygen, hydrogen, hydrogen secretes H2O. then carbon will connect to nitrogen. That H2O, right, the molecule that gets excreted is going to be replaced by the bond. So we can see I've drawn that carbon-nitrogen bond. Water is left on the top. The rest, now you can just draw in normally. So this carbon, double bond, central carbon, variable R group, hydrogen, amine. Nitrogen this side, was it connected to? It lost that hydrogen. It's still connected to one more hydrogen. Then the central carbon, variable R group. So it looked like this. Now, this bond that you have formed here is called a peptide bond. If you are asked to circle it, you'll circle it like this. Do any of you do chemistry by any chance? You do chemistry? Okay. If you do chemistry, you will do the same type of bond in chemistry. However, you'll give it a different name. You'll call it an ester bond. You are not allowed to use that terminology bio. So even though it's the same bond, but you will have a different name for it in bio, a different name for it in chemistry, and you'll need to separate the two subjects out. So this peptide bond that has formed, the reaction that forms the peptide bond is called a condensation reaction. Now, a condensation reaction, right? What is condensation referring to? It's referring to water. So any reaction that releases water is going to be called a condensation reaction. So in fact, even above, when we join together two nucleotides, where is our phosphodiester diagram? Here, right? The phosphodiester bond is formed via a condensation reaction because the OH attached to this carbon is going to combine with the hydrogen attached to this carbon. And that forms H2O, or it releases water. As a real life example, if you've ever been driving behind a bus, you'll notice that a lot of water drips out the back of a bus, particularly if it's parked, you can almost see a pool behind it. That's because the fuel or the combustion reaction that's being done to the fuel is again a condensation reaction. So it's producing water. You won't be tested obviously on fuel and buses in biology, so this is the bond you want to know, but I'm linking it to something you might have seen in real life. Okay. Bonds, yes. Now, if you want to continue the chain, right, so you want to add more amino acid, you can actually do this to either side. So you can add it to this side or to this side because they're both receptive. Obviously, if you're adding it to this side, it needs to be the amine group. And if you're adding it to this side, it's going to be with the carboxyl group. COH. Okay. Any questions on that so far? No? Okay. So we move on. Again, quick summary page. Proteome we discussed at the start. Now, I think these are just some of the easier ones, so that's why I've listed them as a quick revision. Again, some of the easier ones I thought, you can memorize other ones, you can memorize this one, so that's up to you. In terms of like something to be aware of, they might ask you for a non-enzymatic protein. Non-enzymatic protein. The reason they do this is because protein is its own subsection. So if they're going to test that separately later on, they don't want to test enzymes in area of study 1. So that's why I said you should know one example of an enzyme and then one example of another protein that is not an enzyme. Because if they just say protein, you can go with enzymes, but sometimes they will specify non-enzymatic. Okay. So then we're going to move into the protein hierarchy, or how do you classify a protein? There are 4 different ways you can classify the protein, right? Primary, secondary, tertiary, quaternary, all listed here. Primary is just the specific sequence of amino acids joined via what type of bonds? Which bond connected amino acids?
Hydrogen.
Peptide. So hydrogen bonds connected our nitrogenous bases together all the way for here. Nitrogenous bases are connected by hydrogen bonds. But to connect 2 amino acids, right, it was a peptide bond. The secondary structure is going to be referring to the creation of beta pleated sheets or alpha helices. So you can see like the structure, sheets have to have this folding. Now, alpha helix, right? I showed you a double helix structure for DNA. It's got 2 separate strands. Alpha helix is just 1 strand, right? So it's the same strand twirling around. Now, this is what I kind of call the initial stages for folding it. So from this long chain, you've now done a couple of folds, right, into either beta pleated sheets or an alpha helices. So formation of alpha helices or beta pleated sheets. In terms of how they form or what holds those shapes together, that's going to be hydrogen bonds. Right, so those same hydrogen bonds we talked about for nitrogenous bases, they are coming back together now. So in the secondary hierarchy of proteins to form alpha helices and to form beta pleated sheets. Then at the tertiary level, our protein is going to acquire its specific 3D shape. Now, for proteins, right, the shape is highly interlinked into its functionality. Right, your muscles wouldn't work if they were always a round circle. most muscles are very long and thin because that's the shape they need to do like powerful contractions. So if they ever lose that shape, they're not going to be able to do those powerful contractions. And the same holds for every function of enzymes, right? Your immunological proteins, right, need a specific shape to fight infections, to fight bacteria. If they lose that shape, they're not going to be able to fight bacteria anymore. Your enzymes, your hormones, they need particular shapes to bind on to their targets. If they don't have that shape, again, they won't be able to function. So the tertiary stage is where they acquire that specific 3D shape, and hence they will become functional. Now, in this diagram here, you can see as part of that 3D shape, they will have alpha helices and beta pleated sheets embedded within them. So this here is an example of a beta pleated sheet. At the bottom is an example of an alpha helices. If you only have one, right, it has to be the secondary stage. So if you have one beta pleated sheet or one alpha helix, that's at the secondary. But in this example, where I can see both, that must mean it's the tertiary structure. Right. Similarly, if there were like 3 alpha helices and no beta pleated sheets, that's still tertiary structure because there's more than one group. Then finally, at the quaternary level, it has to consist of 2 or more polypeptide chains. So those are our 4 ways, or like we can describe the level of a protein. Now, primary, we said it was joined via peptide bonds. Secondary, we now have hydrogen bonds to join them together. Then what do we have at the tertiary stage? Tertiary is basically every single other type of bond. So hydrogen bonds are going to be there. The one we talked about earlier, so disulfide bridges are going to be there. Now, some others that I will list, ionic bonding is going to be there. Van der Waals forces. You must know hydrogen and disulfide bridges. They're the most important. The other ones you don't really need to worry about because you won't explore them in more depth. I think ionic bonding is a good one to know so that you get 3, which is the maximum they've asked for before. The reason I specifically like to go with hydrogen and disulfide, though, is because hydrogen represents the weakest type of bond, and disulfide bridges represent the strongest type of bond. So they represent both ends of the spectrum. So if you're ever asked how would you want to make a really strong protein, how can you make a protein that doesn't lose its shape? You want to go with trying to use a lot of disulfide bridges, and therefore you would need the amino acid cysteine in that protein. Whereas if you want to go for something weaker, something that's more flexible, right, you're going to go with hydrogen bonds because they're more easily breakable. At the quaternary level, it's the same bonds as the tertiary level because you've introduced all the bonds at the tertiary level. Now, for bio 3,4, proteins become functional at the tertiary level. So at the primary and secondary level, they're considered non-functional or immature, right? They won't work. Technically, in real life, there are some proteins, especially the storage type, that work at the secondary level, but we kind of ignore them in bio 3,4, and we say it's at the tertiary level where functionality comes into play. However, not every protein becomes functional at the tertiary level. Some of them must progress to the quaternary level. Must progress to quaternary. So essentially, what's happening is that you start with your primary structure. Primary will always go to secondary. Secondary will always go to tertiary. Now, at tertiary, there is a check. Is it functional? If it is functional, it will remain at the tertiary level. If it is non-functional, it's going to progress to the quaternary level. And then it must be functional at the quaternary level. So you must get to the tertiary or quaternary to be functional, but the proteins, like it's up to them individually. Will they go all the way to quaternary, or are they going to stop at the tertiary level? Now, I've talked about two terms, right? Protein, and I've also used the term polypeptide chain. Both of these are polymers. Right, so we've discussed that their monomer, we said up here is, oh, I didn't write it down, amino acid. So they share the same monomer. So why are we calling them two different things? There is a very important difference. Polypeptide chains is given, or is a term given to a non-functional protein. Whereas if you say the word protein, the assumption is that it is functional. So that means when I'm talking about the primary structure, when I'm talking about the secondary structure, I must use the word polypeptide chain. But once I've achieved functional protein, right? So for example, the hormone is released into the bloodstream and it's traveling across the body. Or the immune protein is actively fighting against defenses. It's in a mature, formed, and functional stage. That's when it's called a protein. Another way you can think about, is it working? So is it doing its function that it was meant to do out of those 7 functions? If yes, it's a protein. If no, it's a polypeptide chain. Polypeptide chain, again, just to reiterate, is still the chain of amino acids. It just means it hasn't been folded yet into the correct, specific, functional shape. Does that make sense? Any questions? No. Okay. So, a few things that have come up in past Vika questions. I think I mentioned all of these, hopefully. Peptide bonds come from the primary level, hydrogen bonding from the secondary level, all other bonds come from the tertiary level. The protein must, functionality starts from the tertiary level, and the protein must progress to the tertiary or to the quaternary level for it to be functional. It can stop at the tertiary level. It doesn't have to go all the way to the quaternary level. Now, lastly, we have something called prosthetic groups. Now, it might be a little bit tricky to introduce this now, so I will probably recap this in one later, like in the lesson later on. But a prosthetic group is something that, imagine a polypeptide chain intertwined with another type of biomacromolecule. Right. So it might be, say, the amino acid glycine combined with glucose. It's not even necessary for it to have like a sequence of amino acids. It might even just be one amino acid and then carbohydrate, carbohydrate, lipid, lipid. So it's combining some of the other biomacromolecules together. In terms of the tertiary level, though, you must have at least one polypeptide chain. Now, the second or the third, the other chains, they can be polypeptide chains or they can be prosthetic groups. Both of those, right, so a polypeptide chain with a prosthetic group will count as tertiary because it's counted as two separate chains. So primary, secondary, and tertiary, all of these must just be one polypeptide chain and no additions. Whereas tertiary, that's where you have another polypeptide chain or a prosthetic group as well. Now, yeah, I think I forgot to write summary here. This is the summary page. So we've got the characteristics of definitions like primary, secondary, tertiary, quaternary levels. I think it's a different one I read earlier. Hydrophobic interactions. It doesn't really matter which one you choose, as long as you know disulfide bridges and hydrogen, most important ones. Now, those are the bonds that we talked about though. So all of these for tertiary and quaternary, these are occurring between R groups. Right, so your variable R groups. Between R groups. Your peptide bond is between adjacent amino acids, and same for your hydrogen bonds, it has to be adjacent. So if this is an amino acid, they are connected via a peptide bond. If I want to form a beta-pleated sheet, but it has to be with adjacent amino acids like this. But I can't go to an amino acid out here to try to form the beta-pleated sheets because that doesn't really look like a sheet anymore. In fact, this should probably be slightly down. So this is how you can get that structure. However, for your ionic levels, or your disulfide bridges, or your hydrogen bonds, so let me say this is second, then the third or the fourth level, so tertiary or quaternary, but you can have amino acid here, amino acid here, amino acid, amino acid, and then you can have a bond connecting it like this. So they don't necessarily have to be adjacent, and it's via their R groups. Does that make sense? It's okay to say if it doesn't make sense, you want me to repeat anything.
Yeah, I'm a
Like,
bit sorry. I'm a bit confused on the what do you call it? Prosthetic stuff. Like more specifically about the tertiary part. So does it count as is, yeah, no. So like down the tertiary structure. So do
yes.
tertiary structures only form one polypeptide chain, but when it's prosthetic, it forms two? Or is that like I'm a bit confused there on like how many chains it forms.
So at the tertiary level, you can only have one polypeptide chain, no prosthetic groups. Right. So primary, secondary, and tertiary, but you can have a look at these diagrams. This is all one, right? One polypeptide chain, one polypeptide chain. And if you trace this out all the way, it's still one polypeptide chain.
Yeah, but there's multiple like alpha helixes and pleated sheets, like you said, right?
Yes. So if you imagine something like this, right? This is one polypeptide chain. What's happening is you might have some beta pleated sheets here, alpha helices here, beta pleated sheets here. That's from the folding of still one polypeptide chain. So it's going like this, and then it's swirling, right? And it might come back. But if you imagine like you're tracing with a pen, you can trace the entire thing without lifting your pen off because it's one polypeptide chain. Yes, it will have multiple alpha helices and beta pleated sheets, but they're coming from the same polypeptide chain. Prosthetic groups do not apply to tertiary structure. Then when you move on to quaternary structure, right, the example I've given you here is two polypeptide chains. This is by far the most common way. Polypeptide. So you can see they're given in completely different colors. There's one, the second one is slightly behind it. Now, if you are dealing with prosthetic groups, what will be happening is you still have one polypeptide chain. Let's say that's in blue. Now, a prosthetic group, let's draw that in red, is usually going to be kind of small and at the center of the protein-ish. And it might be containing another biomacromolecule. So it can be containing carbohydrates, lipids, but usually not nucleic acids, or even it can be containing like metal ions. So it's called a prosthetic group because its chain is not made up of only amino acids. But imagine it as a polypeptide chain with more things attached to it or with decorations on it. So it's a polypeptide chain combined with some other biomacromolecules. Does that make sense?
It does. I'm just confused on like the structure of a prosthetic. Like if it's like quaternary, tertiary, like
Oh,
it's like
prosthetic groups by themselves do not follow the protein hierarchy structure because it's not an individual protein. Right, it's a compound group, so we don't assign it to a structure.
oh, so it's just a sign. So it's just attached to like a tertiary structure or like a quaternary structure. Is that what you're trying to
Only
say?
quaternary.
Only quaternary. Oh, okay, I got you.
Yes. Any other questions? No. Okay. Again, most of this we should have done. So protein, yeah, it has to be functional, minimum quaternary or tertiary level. I think disulfide bridges we've talked about extensively. So let's do a little bit of practice. This diagram here, but these are MCQ style questions. So basically A, B, C, D would be primary, secondary, tertiary, quaternary. If you're having a look at this diagram, what protein or where are we on the protein hierarchy?
Is it like tertiary, third one?
Yeah. And what do we see that makes it tertiary?
As sheets and helixes.
We do want to use the full name for those. So we want to say alpha helices. You can write the Greek abbreviation as well, or you can write out alpha and beta completed sheets. Now, if you have a look at the diagram below it, what does this one represent?
Quaternary.
Quaternary, good. And what's our reasoning?
It has more than one polypeptide chain.
How many does it have?
4.
Yes. So every time the color changes, that's how they use it as a way to show it's a new strand. They will also use different colors to show alpha helices and beta pleated sheets. So usually they will assign a color for, in this case, they've shown green as alpha helices and red as beta pleated sheets. Those colors can change, but they will always color code it some way, so you can easily tell differences. And last example, what level is this on?
It's tertiary, right?
So again, we can see this was from the booklet above. Uh, Beta pleated sheets and alpha helices. You can also actually see another alpha helices here. So those are some examples of how we typically test it in MCQ. Now, if we have a look at how can they test it in short answer. So here is your scenario. A scientist analyzes a nucleic acid sample and finds that it contains 24% cytosine and 28% uracil. Can you identify the type of nucleic acid? Right, so is it DNA? Is it RNA? And is it going to be single-stranded or is it going to be double-stranded? And then explain your reasoning. I'll give you 1 more minute. Okay, so let's go through the question. The first thing to always look at as soon as you get a question is number of marks. And then the keyword or the command term. So the command term here given is identify. And we also have a second one called explain. If it's a 2-mark question, we can immediately understand that you're going to give 1 mark to each command term. So the command term for identify is pretty simple, right? It's just recognizing the correct type. So the correct one is going to be single-stranded RNA. Now, I mean, that's going to get you mark 1 for command term 2. Now, for the second command term, explain your reasoning. You need to decide why you chose single-stranded RNA. So firstly, you can justify that you chose RNA because the sample has uracil. Now, it's not enough to say the sample has uracil though, right? Why is that relevant at all? So you need to give the link back to RNA, which is unique to RNA and not found in DNA. So now you have justified yourself as to why you picked RNA. You can't just say it's contained uracil. Why is uracil relevant? It's because it's only found in RNA. Then
Question.
you also, yeah,
I thought you said that like it's usually single-stranded, so we shouldn't like assume that it's. If don't you remember like you said like when we were like describing RNA, you said it's usually single-stranded, so we shouldn't like assume it's single-stranded all the time. So I'm a bit confused here.
yes. So we need to justify both parts, right? So far, I've just justified that it's RNA. I haven't justified if it's single or double-stranded RNA. So I also need to justify why I am choosing single-stranded. So we haven't fully answered the question yet. It is single-stranded because of these percentages given, right? If you assume complementary base pairing works, then you should have 24% cytosine, therefore 24% guanine. You should have 28% uracil, and therefore 28% adenine, assuming complementary base pairing. If you add those percentages up, you get 80, 12, 12, 24, 104%. You are not able to get 104% DNA, right? Therefore, complementary base pairing is not at play. If there is no complementary base pairing, remember complementary base pairing occurs between two strands. If there is no complementary base pairing, that implies it is single-stranded. So if you look at the mark allocation, right, which I've written below as well, first mark is simple, just identify the molecule as single-stranded RNA. Now for mark 2, you need to do both things, right? You need to mention there's uracil present, and you also need to show that complementary base pairing suggests it is single-stranded. So as a model answer, right, the unequal percentages of bases are going to suggest it is single-stranded, or you can say because complementary base pairing requires 104% DNA or nucleic acids, which is not possible. So it's a process of elimination, right? You justify that double-stranded is not possible, and therefore it has to be single-stranded. Does that answer your question? Yeah. Okay. Sorry, I did notice the time we already went over. We will leave it here for today. In terms of homework, can I get you guys to read the rest of the response, right? The tips, tricks, especially read the highest scoring answer, and also do the next question. So this question, and then we'll start off with this question next class. At the bottom of the booklet, as I mentioned at the start of the class, there's also an additional sheet for homework. So can I also get you to do this before next class as revision? Okay. Any questions? No. Okay, perfect. Then thank you for your time today. I hope you find this lesson a lot more intuitive and helpful than Unit 1, 2. Most lessons will follow this kind of more now compared to the Unit 1, 2, which is just more revision. And then otherwise, I'll see you guys next week. Thank you.